I made the rounds. I talked to a few people. Some of you picked incredibly technical and difficult subjects to explain, and it looks like you're circling all around it and you're getting it. I have some suggestions in my presentation that might help you as you try to refine. So taking it from there, I have a theory, and it's a theory. You can reject it, but it's my theory. I developed it while I was writing a book about stem cell science, for which I interviewed more than 50 scientists from across Canada and around the world. The premise of the book, which was, I should point out, aimed at a general audience, was to shine a light on the work of Tillum and McCullough, their discovery of stem cells, and their role in developing the field. So the idea was to help Canadians understand and appreciate the enormity of that achievement, something about which most people, most Canadians, are blissfully unaware. So here is my challenge as a writer. The 1961 paper that is now generally regarded as the foundation of the field of stem cells has this earth-shattering title: A Direct Measurement of Radiation Sensitivity of Normal Mouse Bone Marrow Cells. And the follow-up paper from 1963, in which they truly nailed it, cytological demonstration of the clonal nature of spleen colonies derived from transplanted mouse marrow cells. Okay, I come from a newspaper background, and you might notice one thing here. The word stem cells are not in either title. In newspapers, we call this burying the lead. They didn't even mention stem cells in either paper. They called them colony-forming units. So, which brings me back to my theory. Both these beautifully executed papers were written by scientists for scientists. They were written in science. So my theory is that science is a language unto itself. It may be built on English or whatever the scientist's native tongue is. For our purposes today, we'll say English. But it has evolved into a separate and distinct language with its own set of rules and quirks and jargon and meanings and vocabulary. You guys all speak science. You speak it fluently. The challenge is in translating that science into English that ordinary people can comprehend. This is an important skill to have in many areas of your life, beyond speaking to reporters. It will be very useful when you're dealing with academic administrations, hospital boards, useful when you're writing up grant applications. And it'll be very useful in telling your partner or your children what exactly you do for a living. Nowhere is it as more important than when you're trying to explain your work and tell your story to the public via the media. You want to avoid having your story get lost in the translation from science to English. Lisa asked me to provide 3 must-ask questions for you to consider today. These are questions that, if you were to be interviewed by a reporter, would most certainly be asked. So you would need to know, going into the interview, what your answers for these 3 questions would be. They should be top of mind with you. So if you can answer these questions, you're well on your way to translating your important work from science into English. This is not an easy skill, which I'll bring up the concept of MEGO. When I worked at the Ottawa Citizen newspaper, we had a writing coach. That was back in the day when newspapers actually made money and could afford writing coaches. And now they can barely afford reporters. So the Citizen writing coach had a term for boring writing. She called it MEGO. It stood for My Eyes Glaze Over. This was copy that was filled with jargon, long and confusing words, passive verbs, and run-on sentences. They all had a high mego factor. You want to avoid tipping the megometer into the red zone when you're trying to explain your work. And you can do that by explaining your research as simply as possible, by weeding out the heavy science jargon, and using words that people can actually comprehend. Plain English helps people understand, and it helps the media get your message. Now, not all scientists have this skill. Many don't. It's generally not part of your scientific training. I was going to provide some examples of high mego factor scientific writing. But since Trump got elected, I'm trying to veer away from the negative and stay with more positive things. I need to for my own personal psyche. So instead, I'm going to present someone who is really good at it. This is Kim Boycott. Dr. Boycott is a senior investigator at the Children's Hospital of Eastern Ontario and a clinical geneticist there. A few years ago, I interviewed her for a CHR publication about her work advancing gene sequencing, a complicated subject and something of which I knew absolutely nothing and cared even less. So she explained her work so wonderfully well that I was able to pour most of it right into my story. So I don't know if you can read all that, but just to go through it. Gene sequencing used to be a slow, laborious, and costly enterprise. The most popular method, the Sanger sequencing, has been in use since 1977 and in essence involves studying the DNA sequence of one section of a single gene at a time. With next generation sequencing, all 22,000 genes in the human genome can be sequenced in parallel. The process takes 2 to 3 weeks and costs about the same as sequencing a single gene the old way, about $1,000. Consider, for example, floating harbor syndrome, a condition that affects about 1 in 500,000 children who typically are shorter in stature, have difficulty expressing themselves, and tend to have deep-set eyes and a prominent nose. FHS is a difficult to diagnose disorder. FHS is difficult to diagnose because it is sporadic. It doesn't recur in families. Those types of disorders are always more difficult to figure out, says Dr. Boycott. In 2012, using next-generation sequencing on blood and saliva samples gathered from 13 children around the world, scientists isolated the gene mutation responsible for FHS. Since then, they've diagnosed another 50 children worldwide who have the condition. So notice that in those 3 paragraphs, 186 words in total, she answered all 3 of those key questions. Why her work is exciting. She has a way to do something that is much faster and way cheaper. So that's exciting. Why people should care. Because it allows scientists to diagnose a very rare genetic disease and other very rare genetic disease much more easily. And finally, what it means to patients. They don't go undiagnosed. This is, of course, very troubling if you're a parent and you have a child with a disorder that your pediatrician simply can't diagnose. Now they can. So, I should reiterate that most of these words are Dr. Boycott's. She gave me this information. She told her story. She told it really well so that the listener or the reader comes away not bored at all, but actually feeling enlightened, which is what you want to achieve. You want people to come away thinking, I just learned something. There's a side benefit to that, and that is when that happens, the general public thinks, well, our tax dollars are really being spent on good research. So, there are other ways to tell a good story to explain your research. Consider protein expression and signaling pathways. Something very ordinary for you, but very difficult to convey to the general public. Most of the general public thinks protein is what they get in their boiled eggs in the morning, and that's it. That's as much as they think about it. They don't know anything about proteins and enzymes and signaling pathways. And to be perfectly honest, I barely do. I have to check it and have it explained to me by scientists all the time. And I write about science for a living. So here's how Dr. Paola Marignani explained her work on LKB1, which is a protein linked to several kinds of cancer. She said, if you lose LKB1 or it's mutated, it sets up protein signaling pathways for disaster. It would be like an intersection that once had stop signs and is now without any. Eventually, an accident is going to happen. So Dr. Marignani used this analogy to explain what happens to LKB1 if it's missing or mutated. Everyone in this room could argue that protein expression has absolutely nothing to do with traffic signs. I know you'd be right. But her analogy is brilliant because it conveys a very complicated message in terms that anyone can understand. It tells a story that people can follow. It works. So while we're on the subject of storytelling and telling a story well, you might want to consider the importance of specifying. Generalizations generally don't work. Skipped one. Oh, I believe that. If I tell you a hockey game I went to last night was a great game, or it was amazing, or it was awesome, I've told you nothing. If I tell you the hockey game was great because the Ottawa Senators goalie Craig Anderson stopped 45 shots from the Leafs, including 3 breakaways, you have a much better feel for the game and why I thought it was a great game. Specifics work better than generalizations. Generalizations have a high Mego factor. I recently profiled a man named Dan Muscat who underwent a bone marrow stem cell transplant for systemic scleroderma, an incredibly painful condition. I described his pain as intense. The word intense doesn't really convey much. What picture does it paint? What does it mean other than bad? So I got out of the way and I let Dan Muscat describe the pain of scleroderma. He said, it's like hitting your finger so hard with a hammer that it makes you want to put your hands between your legs and cry. That is an absolutely wonderful description of excruciating, intense pain. And it paints a picture. When you're talking to reporters, when you're trying to explain your work, think of ways that you can illustrate it that convey a story, that people can latch onto, that people can get, that the media can understand. It conveys the meaning quickly, which is also very important, because when you're explaining it to the media, they really don't have much time to spend listening to you. As Ivan mentioned earlier, the media is not in such great shape. Across North America, both in print journalism and in broadcast, newsrooms are being cut back. In many newsrooms, they're down to skeleton crews reporting everything. The old media is particularly hurting. When I was city editor at the Ottawa Citizen, we had a staff of 30 reporters covering local news. That was just local news. And we're not talking about sports, entertainment, arts, the Hill. That was 30 people covering local news. We had 2 health reporters, 1 science reporter. We had 2 education reporters, 1 for each board. That situation just does not exist anymore. There are very few papers in Canada that can actually afford to designate a reporter as a science editor. The Globe and Mail is an outstanding example of a paper that has an outstanding science reporter. This is not something most mainstream local newspapers can afford, and certainly not many broadcast outlets. So the point is, they're stretched very thin. And instead of specialized beat reporters covering specialized beats like education, like science, they're mostly general assignment reporters covering absolutely everything. So the reporter who shows up at your lab to interview you, and that's actually a fallacy, because a reporter actually showing up at your lab doesn't happen anymore. They call you. It takes too long to get to your lab and too long to get back, and they're on deadline. So probably they'll phone you. But a broadcast outlet would send a camera, of course. Anyway, very few newsrooms can afford to have a designated science reporter, someone who is up to date and well-versed in various areas of science beyond yours. So that's what you're up against. You have to explain your science to these overworked reporters who probably know nothing about the science you hold dear. And you have to explain it in a compelling way that they can comprehend. And again, this is a general assignment reporter who was yesterday covering a traffic accident. At max, you've got 20 minutes. If you can do that, if you get that, you're lucky. You need to be prepared to tell your story quickly and clearly and in terms they can grasp, and you need to minimize or at very least explain the heavy-duty science terminology that you use every day in your job. So, skipping ahead, because I see I only have, yeah, there's my little cartoon on the media. I don't know if you can spot that. So, in terms of jargon, here are 20 terms that stem cell scientists use all the time that mean almost nothing to the general public. You guys eat this stuff for breakfast. It makes perfect sense to you. But to your man on the street, your woman on the street, they have no idea what you're talking about. And you have to be prepared to explain all this to a reporter who shows up at your door or calls you on the phone. I've proposed here some possible explanations that might work for you. They might be oversimplified. This is just how I would try to go about it. But I have scientists all the time talk to me about, we're working with animal models. Just say animals. No one understands autologous. You have to explain that that's the donor's own cells. Why would you say decreased mortality? Why not say fewer deaths? Fewer people died. Differentiation is a basic tenet of stem cell and regenerative medicine, but you have to be prepared to explain that. Hematopoietic, whew. Heterogeneous, forget about it. And I think I mentioned there, Boolean, don't even go there. Here's a few more. High throughput screening, mesenchymal or mesenchymal, mouse models, just say mice. Pathology, the causes and effects of disease. Pluripotent, can make many different cell types. Progeny, sons and daughter cells. And my favorite, xenotransplantation, which I always have to check, even though I'm pretty sure I know what it's about. I'm not saying these words should never be used. Your science is your science. But what you have to be able to do is explain what they mean to a reporter whose eyes are glazing over. Finally, because I think I'm just about out of time, a word about honesty. Reporters balk at being sold a bill of goods. And one of the sins that I think the stem cell community committed in the early days was overselling the product, so that there was a lot of hype being pushed out. Reporters are really tired of being oversold and hyped. It can really help your story if you just tell the truth. Don't say something is a breakthrough if what it is an incremental change. You can be excited about how neat it is and what it does, but don't oversell. I will conclude with a quote from Harry Atkins, this guy's a hero of mine because he's doing great work with bone marrow stem cell transplants on a number of autoimmune diseases. He was Dan Muscat's doctor. And here's what he said. It's going to take months and months to know if this is a real improvement. The changes with scleroderma don't happen overnight. This is not a curative treatment. It doesn't make scleroderma go away and never come back. It's something that helps the population of scleroderma patients, and some patients improve, some do not. Honesty is incredibly refreshing. So thank you very much. That's what I've got. If there are any questions or I can help you in any way, just let me know later. Okay? Thank you.